Today our topic of discussion for Standard 12 is Mendels law and the variations of different laws of the Mendel. Now in 10th standard you have already studied about the Mendels law, the basic of law of dominance, law of segregation and law of independent assortment. Now here we are going into more details of these various laws. Now we'll just make a revision of what is a phenotype, what is a genotype, what are alleles, all these things, because these terms we require again in the topic of the discussion. Now phenotype generally tells us about the external characteristics which are expressed by the genotype or the gene type. Genotype is the basic genetic constitution which occurs in pairs, and as there are diploid and haploid organisms also, Mendel's law is applicable mostly in the diploid organism, but there are variations of it. There are exceptions to the Mendel's law, and both law of dominance and law of segregation, law of independent assortment, these are known as the intergenic and intragenic gene expressions, which we studied later on. Now, usually we say in monohybrid cross, we cross in a plant with one character. Now the character here is, let us take for example the tall plant and the dwarf plant. In the parental generation we have a pure tall, which is expressed by capital T and capital T, and a pure dwarf, small t and small t, which are called as known as pure lines. And in the F1 generation we get a tall and a dwarf gene, because during the process of meiosis, when they form the gamete, the capital T gene or the factor comes from this and small t comes, and it is expressed as a hybrid tall. Now we do selfing. There are various types of cross. Self crosses are there, test cross is there, back cross is there, and reciprocal cross is there, which will come to the later stages. But let us know, selfing means crossing with a similar genotype. So when we cross with a similar genotype, we express this in form of Punnett square, where this two forms gamete, capital T small t, and this forms capital T small t. And when they fertilize, they have this type of combination. Capital T gives tall, which is a pure tall, capital T small t hybrid tall, capital T small t hybrid tall, small t a dwarf variety. Now this is expressed as we say this is a phenotypic expression. The expression between tall to dwarf is 3:1. So phenotypic means externally what do we see? Externally we see there are tall, but among this tall, 1 is pure, 2 is a hybrid tall, so that makes 3. So the ratio is 3 and 1 dwarf, so 3:1 we see. But when we know the exact genotypic expression, we say pure tall is to hybrid tall is to dwarf is equal to 1:2:1. Now this is the monohybrid cross. We have several examples of the monohybrid cross. We can do with all the 7 pair of characters that were done by Mendel on the plant Pisum sativum, like say for the seed shape, seed color, pod shape, pod color, all these different other factors. Now we will see that from this, the law of dominance, the first law is the law of dominance. Out of one pair of allelomorphic character, one is dominant and the other is recessive. That means in this there are two factors which we call the T and T. These are the alleles, and small t and small t, which expresses dwarf. Now when they combine the capital T and small t, these are several factors or the several genes. These term genes were turned by Johansen in 1909. So here the tall gene is suppressing the dwarf gene. So this is expressed as a tall. Now This is the law of dominance. Law of dominance says that out of two allelomorphic character, one is expressed and that is said to be the dominant, and one is the suppressed, which is the recessive characters. But this is applicable to Mendels law as because all the genes that we study in Mendels law are non-linked genes. If there is a linkage, a complete linkage or an incomplete linkage, we don't get this, and that we study in all the expressions, the intragenic expressions, which we see that does not comply to the Mendels law of dominance, neither comply to the law of segregation and independent assortment. The second law says the law of segregation or purity of gametes. Now in this law, again this we derive from the monohybrid cross. So capital T, small t, recessive, and this is the dominant and the recessive. The pure line gives capital T, small t. When self cross gives this ratio 1:2:1. So 1/3 is pure tall, 2/3 is hybrid tall, and 1/3 is pure tall. Now what is law of segregation? What does it state? The law of segregation states, or what we call the law of purity of the gametes, that in the first generation, when we see the F1 generation is pure tall, it is thought, or it can be, we can think that the dwarf gene has vanished. But did it really vanish? No, it did not really vanish, because when we self cross it, the reappearance of the dwarf gene here, it states that this gene must be there in a suppressed condition. So, the reappearance of this is a proof that when the gamete formation is taking place, then the gametes remain pure. That means they do not blend together to give certain other characteristics. But again we say this is applicable to all the Mendelian factors where there is a linkage, where there is incomplete or complete linkage. There are exceptions like incomplete dominance. We'll study that later, or codominance. We'll study that in later chapters. Now let us go to the law of independent assortment. For this law we have to go to the dihybrid crosses, because law of dominance and law of segregation is derived from the monohybrid cross, but the dihybrid cross gives from the dihybrid cross we generate, or we formulate, or the Mendel has formulated the law of independent assortment. Now here we will see that what is the dihybrid cross. In dihybrid cross we take two characters. Now one character is round, which is expressed as capital R, and yellow. These both are dominant, and smaller, which is wrinkled, small y, which is green. So round is dominant over wrinkled, yellow is dominant over green. So when we see that they take a pure line of two different characters at a time, then the gamete formation from here will be capital R, capital Y, because from each of the factors will come in the gamete under the process of meiosis. Here small r and small y. So when they fertilize, they give rise to capital R small r and capital Y small y. That means when it is expressed, we see according to the law of dominance individually, R is suppressing small r, so it will be expressed as round. Capital Y is suppressing small y, that is yellow is dominant over green, so it will be expressed as yellow. So in the F1 generation, we see all seeds are having round and yellow phenotypically expressed. This is done by, now what we do is, like in monohybrid cross, as we have done the selfing, again we do the selfing. So when we are doing the selfing, let us see what we do in the selfing. Okay, now in selfing means we are crossing with the same type of genotype, that is capital R small r, capital Y into capital R small r into capital Y small y. That means the same genotype is being crossed. Now when this genotype will form the gametes, so what are the combination? Capital R and capital Y will come here. Small r capital Y, capital R small y, and small r small y. All the 4 different combinations. Okay. Now say in this case of, say, in the female gametes, in the ovum, or what we call the female gametes, capital R capital Y. Okay. Then small r capital Y, capital R small y, and small r small y. Now these are the different gamete formation, and these are the male gamete and these are the female gametes. Now when they fertilize, these are the combination which arises. Now if we see this different combinations, then we see that how many round and yellow are there? There will be 1, 2, 3, 4, 5, 6, 7, 8 and 9. So 9 round and yellow. Then round and green will be 3, wrinkled yellow will be 3, and wrinkled green will be 1. So we say that phenotypic expression of the F2 generation is in the form of 9:3:3:1. Now let us see that how it will be expressed once more in the book. So what are the combination of the F2 generation? Round and yellow, that will be 9. Round and green, that will be 3. Wrinkled yellow, that will be 3. Wrinkled green, that is 1. So that means the phenotypic ratio is round yellow is to round green is to wrinkled yellow is to wrinkled green is equal to 9:3:3:1. Now this is what we call the phenotypic ratio of a dihybrid cross. Now what is the genotypic ratio? Genotypic ratio will be various, because see, this is also round yellow, this is also round yellow, this is also round yellow. It is quite a complex form. So the genotypic ratio is a more complex form. So we say that what is the law of independent assortment? Independent assortment means when there are 2 pair of characters are crossing together, these 2 pair of characters do not interfere into each other. That means do not blend. The characters, when they are segregating and uniting again, they do independent of each other. How do we know that? We know that because we see here that these 2 characters, these are the parental characters which we started with, that is round and yellow and wrinkled and green. And these 2 are called the recombinants or the variants. So the parental generations are there, which are in form of round yellow and wrinkled green, and these 2 becomes the recombinant, which are the new or the variants. Now that is what we say independent assortment, that when 2 characters are crossed together, these 2 characters do cross and form the offsprings. These offsprings are expressed independent of each other and they do not blend.